This method can facilitate research in the tissue engineering field related to tissue development, cell alignment, and mechanical analysis of polymers and engineered tissues. The main advantage of this technique is that it is low cost, easy to use, and fast.
We developed this technique in my lab because geometric control of engineered tissues on the millimeter to centimeter scale was not being done in the field, and we saw a critical need to think translationally about scaling up the size of engineered tissues. This technique allows us to leverage the benefits of geometric cues on cellular physiology in engineered tissues through fabrication techniques using posts and a variety of shapes.
This method can provide insight into the production of engineered tissues with controlled size, shape, and alignment. It can also be applied to various disease models, personalized regenerative medicine, and for the translation of engineered tissues to preclinical studies.
Begin by opening a vector graphics program on a computer. To prepare the desired mold geometry in vector format, open a new canvas of appropriate dimensions with the RGB color format. Then, select the Shape tool from the left-hand panel and create the desired geometry by entering the dimensions at the top of the window to precisely define shape sizes.
Design the mold geometries to allow for at least a 6 mm border between the edge of the outermost features and the cutting line to permit trimming of the PDMS meniscus following mold casting. This will allow the finished mold to lay flush with the bottom of the culture dish.
Next, open the color picker in the top left-hand corner of the window and define new color swatches that are compatible with the laser cutter software. Select each path and assign it an appropriate color from the cut swatch. When finished,